So as people are joining in, I just wanted to mention that we have a little secret here. Everything we've done so far as part of this series has been recorded and is available on the website. And if you go, we want to find it. The best way is RC Wiki. You type RC Wiki once you're on the intranet, and this will take you to the research computing website. And under digital science education, you think you would go here, you would find that right now it doesn't work. You have to go on the left-hand side, and that's repeated here, digital science education. So you just click on the very left, digital science education. This will take you to the page that Shira's group developed. So we have upcoming seminars, including today, May 26th, Tomasz Nowakowski. Then next week we have the speaker, University of Cologne, and this MGH World Collaboration. We'll talk about, the speaker will talk about the identification of pathogenic variants and rich regions across genes and gene families. And then Andrew Sharp from Mount Sinai School of Medicine will talk about this BioRXiv preprint on June 23rd, survey of rare epigenomic variation in 23,000 human genomes. So those upcoming seminars, we're planning for more, but whatever has been finalized will be posted on this website. If you scroll down, you can watch embedded videos of our previous presentations. So again, I encourage you to go to RC Wiki, and on the left-hand side, on the digital science education, you will have the page of everything that has been so far presented, and as well as list of the upcoming seminar. And as you know, if you go back to Research Computing Wiki, there's other interesting resources that you can benefit from to support your research, in particular remote genomics, bioinformatics, all those areas. So we'll be starting in, let's give another minute before we fire up. All right, so again, under digital science education, we have the page with all the resources for this series, including previous talks. So today we have Jill Madden, and is going to take over right now and introduce the speaker, and we'll fire up our presentation in a couple of minutes. So Jill, I'll just pass it to you. And,
great, thanks,
you can
Piotr.
introduce the seminar.
Yeah. It's my pleasure today to introduce Tomasz Nowakowski, who comes to us, is zooming in from the University of California, San Francisco. He did his training in the University of Edinburgh for his PhD, and then came to the University of California, San Francisco for his postdoc, and is now assistant professor there, and looking at the developmental origins of cell type heterogeneity in the human nervous system. And he has a preprint available in the BioRXiv titled Single Cell Epigenomic Atlas of the Developing Human Brain and Organoids. And he'll be sharing his work related to that paper and others with us today. So thank you so much, Tomasz, for taking the time to share your work.
Absolutely. Thank you so much for inviting me. It is a pleasure to give this talk, and I thought I would start sort of by introducing the broad topic of interest to my and other labs, which is the human cerebral cortex. Can you see my mouse pointer just to make sure?
Your presentation is not shared yet, so.
Let me try to fix that.
Just as Tomasz is setting up, we'll be taking questions at the end, and we'll allow you to unmute at the very end so you can ask directly, or alternatively, you can pass it in the chat window to me. We've typically people ask questions directly at the end to kind of.
Okay, great. Can you see my screen now?
You can see your screen right now.
And can you see my mouse pointer?
We can see your mouse, and we can see you as well.
Awesome.
We got it all.
Perfect. Okay, we're all set. Yes, so as I was saying, my lab is interested in the human cerebral cortex, which is this outer shell of the human brain. It's a sheet-like structure that contains billions of neurons distributed across dozens of functional areas that is responsible for many of the higher order cognitive functions in humans. So there is a lot of interest in trying to understand how these neurons are unique and similar across functional areas to support these diverse functions. Even though the cerebral cortex is this sort of semi-uniform sheet, which we often sort of refer to as the 6-layered cerebral cortex, whenever you're looking at different functional areas or anatomically defined areas, you will actually find a lot of variation. It's not true that the cortical sheet is uniform. If you look in the visual cortex, you will find multiple sublayers of layer 4. If you look at some of the more frontal areas, you will actually almost not recognize layer 4 at all. So there's quite a lot of variability in terms of the organization of these cells that's been appreciated since the days of Santiago Ramón y Cajal. What underlies some of these differences includes different populations, different types of neurons with distinct patterns of connectivity. For example, the two major subtypes or subclasses of neurons that are found in the cerebral cortex include excitatory neurons and inhibitory interneurons. So excitatory neurons are the primary signal generating cells, and those cells communicate with each other as well as with the inhibitory interneurons, which form these microcircuits within cerebral cortex. And whenever you look across different functional areas, these patterns of circuits and composition will be different, which underlies the sort of the fundamental function of cortical circuits, which is to receive information, process it in a certain way, and pass it on to another area. And however, the cortical neurons share developmental origin. And when you look in development, both the histology as well as the basic patterns of neurogenesis and neuronal organization appear much more uniform across the prospective functional areas. So this is a cross-section, a parasitical cross-section through the human brain around mid-gestation from an anatomical atlas. And as you can see, the anatomy is very uniform across these cortical areas. And whenever you look in a single area, you'll find this pattern, shared pattern of radial glia, which act as neural stem cells, differentiating either directly into neurons or into intermediate progenitor cells, which are neuronally committed progenitors that give rise to neurons. And neurons that are born in the germinal zones then migrate into the prospective cortical plate, where they organize into layers. And so, as I said, this pattern of neurogenesis seems to be conserved across areas. In contrast, the inhibitory interneurons originate from the ventral telencephalon, or a region in particularly called the ganglionic eminences. And these cells are born and pattern in response to developmental signaling pathways originating in the ventral telencephalon, and then they migrate tangentially to populate the cerebral cortex. Many of these processes are vulnerable to mutations and environmental perturbations, and many of those have been recognized into and classified into different syndromes or disorder types, including both neurological and neuropsychiatric disorders, which affect approximately 1 in 6 children in industrialized countries. So this is quite a big problem, and therefore understanding of how these mutations might affect normal brain development are so crucial to our understanding of these disorders and development of new treatments. Whenever we talk about cortical development, I always like to think back to the sort of fundamental units in biology. Most of these cells, or all these cells, share the same, pretty much the same genome, and yet they develop into such different types of neurons, such different types of cells. So how is the activity of the genome regulated to give rise to these different populations is the central question that developmental biologists seek to address through increasingly complex technologies. And in the cerebral cortex, especially, tremendous amount of work has been done over the past decades to recognize or identify distinct subtypes of both excitatory and inhibitory interneurons. Work, for example, through fantastic investigators such as Jeff Macris and Paula Arlotta have identified a number of transcription factors that regulate subtype specification in cortical excitatory neurons, as well as work from Gord Fischel and others recognizing subtypes of interneurons and how and where they are originated. So quite a lot of this subtype diversity has been defined, but the developmental origins are still quite unclear. Many of these signatures, including layer markers, are conserved across species. And this is thanks to the fact that the origin of especially cortical excitatory neurons is very highly conserved. The developmental history is very highly conserved across species, such that radioglial neural stem cells undergo sequential and progressive generation of more and more superficial cortical layers, as shown over here in a panel derived from Pashkorakic's work doing birth dating experiments in macaque monkeys in the 70s. So as you can see, each of these bars represent a position of a labeled neuron during development by injection of thymidine derivatives at different days of embryonic development, and where these neurons are found in postnatal life. So this pattern of inside-out generation of cortical layers is very, very highly conserved across evolution. But what about functional areas, which is a topic that I alluded to earlier in my talk? When you compare mouse and humans, one of the striking differences is that in general, mouse cerebral cortex has fewer functional and anatomical areas compared to the human cerebral cortex, which has many more. And so there has been a long-standing challenge or request to try and identify how these developmental areas, how these areas emerge, and what are the molecular correlates of their emergence. And some of the early work was done using microarray transcriptomics. This is a panel taken out of a fantastic paper put together by Ed Lane's team at the Allen Institute for Brain Science, where they were able to microdissect different areas of the human brain, including both cortical areas highlighted over here with a red square, and also subcortical areas, cerebellum, and so on. And what you're looking at here is a similarity matrix where the heat map indicates the number of differentially expressed genes between any two structures in the human brain. And as you can see, whenever you compare cortical areas to any of the subcortical areas, or almost any subcortical area to each other, you will find a number of differentially expressed genes. But when you consider the cerebral cortex, you actually, using this sort of very old school bulk transcriptomic approach, you find very, very small number of differentially expressed genes. So at the molecular level, with bulk tissue transcriptomics, you find very, very subtle variation, almost no variation at all. And so what establishes the pattern of arealization is the physical location. Over here to the right, it is a depiction of the developmental organization of cortical arealization taken from or inspired by Pashkorakic in the 80s, who proposed the radial unit hypothesis and the protomap hypothesis, such that every sort of region of the germinal zone in the cortex projects directly onto the postmitotic region in the cortical plate, and that there is a one-to-one correspondence between a region in the ventricular zone and a region in the cortical plate where neurons deposit, such that any positional identity that's encoded in the germinal zone in progenitor cells gets projected directly onto the identity of neurons that are derived from that proliferative unit. So rather than having to sort of invent molecular regulators of regional identity, you're simply projecting this entire layer onto one another. However, over the years, research from a number of labs has identified several key developmental signaling factors, signaling centers, including the cortical hem, the anterior neural ridge, and also the floor plate, releasing developmental signaling patterning factors such as the BMPs and WNTs, the fibroblast growth factors, and the sonic hedgehog, which diffuse through the tissue to establish gradients of these pathways. And the neural stem cells or the radial glia, which sense different combinations of these factors, then respond by expressing patterning transcription factors in different, which are also expressed in gradients across the sheet of the neuroepithelium, including PAX6, EMX2, SP8, and COOP-TF1. And several of these transcription factors have been studied extensively in mouse and have been shown to have regional patterning defects. But this is still quite different to the, from the sharp boundaries that we recognize in the adult animal, where both gene expression differences can sharply change between, between functional areas and guide connectivity development. So how do we go from these gradients of transcription factors to distinct or discrete boundaries between functional areas has been very much unclear until the work from Axel Wiesel and Len Penacchio, who have been identifying and testing a number of enhancer elements that are identified through epigenomic studies. And what was remarkable using these stable transgenic animals for activity, testing for activity of these enhancer elements, is that unlike transcription factors and gene expression profiles, enhancer activity can be very, very restricted to distinct areas of the cortex. So foreshadowing this idea that perhaps enhancer activity, enhancer code might be underlying this or foreshadowing the emergence of functional areas. Why is this interesting? Well, there are a number of transcription factors and genes, including OTS2, which are actually differentially expressed between the prospective functional areas in the cortex, which have been implicated in neurodevelopmental psychiatric disorders. Moreover, when you look at the whole transcriptome analysis and perform an analysis of differentially expressed genes between different brain areas and throughout development, and look for the enrichment of genes implicated in psychiatric disorders such as autism spectrum disorders, you'll find an enrichment for genes mutated in autism in the prefrontal cortex in particular. Moreover, if you look across evolution, while several of the genes that are differentially expressed across areas are very highly conserved, such as calbinin 2 over here, there are other genes that completely change across evolution. For example, neuropeptide Y, which is expressed in the occipital cortex in the human and in the frontal cortex in the mouse. So essentially what this tells us is that the areas or the areal patterns of gene expression could be really important both for understanding the functional organization of circuits, but also could be tremendously important if we want to understand the etiology of neuropsychiatric disorders. So to address some of these questions more extensively and revisit some of these questions, several years ago, we've been I've been lucky to work with a tremendous team as when I was a postdoc in the Krickstein lab, working with Alex Pollan and folks at Fluidigm Corporation to leverage the access to some of the early single-cell transcriptomic approaches. And what we did was we took advantage of our ability to access primary human fetal tissue samples from second trimester of human development. And in some cases where the tissue is more intact, we were able to actually microdissect or identify specific functional areas, including the prefrontal cortex, the visual cortex, and the medial ganglionic eminence, which is the major source of cortical interneurons. And using single-cell transcriptomics and unbiased bioinformatic approaches, we were able to identify molecularly defined subtypes of each of the cell types that we sort of care about, both identifying the excitatory neuron lineage as well as the inhibitory interneuron lineage. And something that I won't go too much into the details of, something that was quite remarkable was when we projected the anatomical area of origin information onto this map. Something that appeared very surprising to us was that when we looked at the origin or anatomical origin of excitatory neurons, pretty much all of the clusters of excitatory neurons were organized according to the area from which they were sampled. This was not true for inhibitory interneurons or radioglia cells or intermediate neural progenitor cells located over here. And we validated differentially expressed genes using immunohistochemistry, as well as in situ hybridization. And something that was extremely striking in that example was the shared pattern of co-expression of BCL11B and SADB2, which are two transcriptional regulators which direct different projection patterns of excitatory neurons towards the pyramidal tract or callosal projections. And these two genes, these two transcriptional regulators, are co-expressed in prefrontal cortex excitatory neurons, but they are differentially expressed in excitatory neurons in the visual cortex. We later went on and also validated or extended this analysis together with excellent postdoctoral scholar Aparna Baduri in the Krickstein lab, as well as two folks in my lab, Ryan Zifra and Chang Kim, who took advantage of the emerging higher throughput technologies with 10x genomics to repeat some of those experiments using a higher throughput method. And the pattern that emerged was very similar, such that the neural stem cells or radioglia down here are essentially grouped together in the same cluster. And the same is true of intermediate progenitor cells, but the differentiating excitatory neurons sort of seem to develop into distinct subtypes. This was later also confirmed by work done in mouse by Basilka Tasik at the Allen Institute for Brain Science. And what the group at the Allen Institute did was they sampled cells from the anterior lateral motor cortex, which is the closest proxy for the mouse prefrontal cortex and the visual cortex. And when they compared using single-cell transcriptomics the signatures of excitatory and inhibitory interneurons, they found almost exactly the same pattern, such that inhibitory interneurons identities were shared across these two areas, while excitatory neurons are distinct according to the area from which they were sampled. Of course, this is only one one pattern which we discovered, but it was a very striking one because it suggested that distinct cell types emerged in these different areas of the cortex, in these different topological domains of the developing tissue. And of course, as a developmental biologist, I am most interested in trying to understand how these patterns emerge. And one of the most fundamental questions is whether there are distinct transcriptional or epigenomic regulators that promote differentiation into these different identities, as demonstrated here in a diagram drawn by Conrad Waddington. So we're really interested in looking at this map underneath the map of transcriptomic differences underlying differentiation patterns. And to address this question, we've taken advantage by a technology that was initially developed by Jason Buenrostra in Will Greenleaf's lab, which was termed the assay for transposase accessible chromatin, which takes advantage of barcoded TN5 transposase, which can integrate into the open chromatin region and introduce sequenceable barcodes or oligonucleotides. And the property of the TN5 transposase is that it will integrate more preferentially to sort of these relaxed parts of the genome initially before sort of starting to open up the heterochromatin. And so that allows you to sort of identify regions of open chromatin. And this is really the technology that was first adapted for single cell application, and with the commercialization by 10x Genomics, this really was democratized and scaled to the level where you could start to revisit some of the same questions that we've asked before in primary human fetal tissue. So with that technology in hand, Ryan Zifra in my lab, together with Chang Kim, a bioinformatician, profiled single cell or single nuclei in this case from distinct, isolated from tissues from distinct anatomical areas of the brain, including some of the same areas that I've mentioned before we profiled with transcriptomics, but also the medial ganglionic eminence, which is the major source of interneurons. And after purifying nuclei, they profiled samples, 6 biological replicates of samples from around mid-gestation, and performed single cell ATAC-seq. Of course, as with most of these single cell genomics approaches, when you profile samples individually, you will get quite a lot of batch effects. But there are now bioinformatic approaches, including SCL9 from Gerald Kwon's lab, that allow you to correct for some of those batch effects. And as you can see, pretty much all the clusters that we've identified in our analysis had contribution of cells from pretty much all individuals. We could not identify significant contribution of either sequencing depth, fraction of reads fragments in peaks, or even a substantial contribution from the type of sample that was processed, be it either a fresh or a frozen sample, and both of those went into our analysis. Moreover, aggregate of single cell ATAC-seq reads correlated very highly with bulk ATAC-seq performed in parallel. We used an analysis pipeline developed by Bing Ren's lab, Rongxin Fang, called SnapAttack. And what that analysis pipeline does is it takes your reads, aligns it to the genome, and splits the genome into 5 or 10 kb bins, which allows you to perform both feature selection and similarity analysis by using Jacquard distance, perform normalization, and clustering. And using this approach, we were able to then perform both clustering and post hoc peak calling to identify peaks. One of the advantages of this method is that the peaks are very often correlated with gene expression, such that when you look at peaks present in promoters of different genes, you can use that information as a proxy for gene expression. So in this analysis, we've identified, or we've filtered out high quality cells, which left us with approximately 80,000 single cells, single nuclei, which clustered into, I believe, 25 or 26 molecularly distinct clusters. And when we projected this gene activity, as some people refer to it, information against this map, we could clearly identify some of the same populations as we've seen with transcriptomics. including radioglia neural stem cells characterized by the expression of glial fibrillary acidic protein, GFAP, which does not overlap with eomysodermin, which is a transcription factor identifying neural progenitor cells, NeuroD2, which is a transcription factor marking excitatory cortical neurons, and DLX1, which marks cortical interneurons. So the expression or the predicted expression of these genes did not overlap, instilling confidence in our analysis. We then took advantage of the fact that previous single-cell transcriptomic approaches have defined for every one of those cell types a set of genes that are characteristic markers. And by correlating, in this case, gene activity scores inferred from single-cell ataxic with cell type specific genes identified in single-cell transcriptomics, we were able to project or infer the molecular identity of every single ataxic profiled cells. And this map made a lot of sense, such that cells that were most highly predicted to express GFAP were identified as either astrocytes or radioglia cells, EOMS positive intermediate progenitor cells, NeuroD2 positive excitatory neurons, which split into two major clusters, deep layer neurons and upper layer neurons, and then also cortical interneurons. And interestingly, samples profiled from medial ganglionic eminence clustered over here and actually shared or projected most highly to radioglia and intermediate progenitor cells present in that region. And those cells clustered away from neurons, interneurons that were profiled directly from cortical tissue. So you can think of those as being the progenitor cells of these cells over here. And so overall, this really gave us the confidence that we're looking at a map that's high quality and makes a lot of sense. Using activity by contact model, which was developed by Jesse Engreitz, we applied this algorithm by taking advantage of the ataxic data set that we generated, as well as cut and tag data generated for H3K27 acetyl mark, which is a marker of active chromatin, previously published high-C data from Dan Geschwin's lab, and also gene expression data sets that were aggregated from single-cell transcriptomics. And what this algorithm does is it actually allows you to predict interactions, long-range interactions between gene promoters. For example, over here, you have a coding locus for NeuroD6, which is a transcription factor that promotes excitatory neuron differentiation, and long-range predicted interactions with distal putative regulatory peaks, some of which are marked, or many of which are marked by H3K27 acetyl marks, giving us sort of a window into these putative regulatory elements. We were able to intersect our data sets with published both roadmap epigenomics data sets, especially the Capstone consortium, to further sort of validate and provide further to further validate our predictions of these regulatory elements, as shown over here, intersection with both fetal brain putative enhancers from Capstone. We also intersected our data with PsycEncode, and in those data sets, you have two different types of intersections that you can do. You can either intersect the data with adult tissue, which in adult tissue enhancers were actually enriched for enhancers present in astrocytes and radioglia cells, which makes a lot of sense because the adult human brain is quite full of astrocytes, whereas intersection with fetal data sets showed enrichment in neurons, which again makes a lot of sense because before there are any astrocytes in the developing cortex, they're primarily neurons. So the enhancers that are identified in those data sets seem to be enriched for excitatory neurons. You can also perform a very nice analysis where you can look at the presence of previously validated VISTA enhancers, and a lot of these previously validated enhanced forebrain enhancers overlapped with or formed a subset of the peaks identified in our analysis. It is also very interesting. One of the analyses that we sort of like to do was to overlap our fetal peaks with human accelerated regions, some of which are thought to underlie brain evolution. And indeed, we had a nice overlap of about 900 human accelerated regions that overlap putative regulatory peaks. And when we looked at the enrichment of these peaks, we actually found a pattern where most peaks were sort of enriched in neural progenitor cells, both in the medial ganglionic eminence and in the cortex, suggesting that perhaps these populations might represent some kind of hotspot for evolutionary activity, which would be really interesting and worth examining. But another, a more practical implementation of this data set is to be able to identify transcription factor code that underlies cell type specification. And by taking cell type specific peaks, we are able to calculate motif enrichment, perform motif enrichment analysis, and identify transcription factors whose binding motifs are overrepresented in cluster-specific peaks. And again, this analysis made a lot of sense. For example, clusters 22 over here and 3 are actually cells that were derived from medial ganglionic eminence, and they're enriched for NKX 2.1, which is a transcription factor that specifies medial ganglionic eminence. And the same was true, especially for interneurons, but also for some of the excitator neuron types. So a lot of this analysis reassured us that the peaks that we are identifying do exhibit these pattern of transcription factor enrichment that we would expect. We also wanted to look at reorganizing this map of transcriptomics and epigenomic landscape underlying differentiation of excitator neurons in a shared map. We performed an analysis where we co-embedded single-cell transcriptomics and single-cell epigenomics in the same space using single-cell align and multi-CCA. And this analysis organized both transcriptomic and epigenomic signal in the same shared map with some divergence, which is to be expected because these modalities are so different, but nonetheless, it organized cells along a pseudo time analysis. And what that allows us to do is to then identify peaks that are differentially accessible across differentiation trajectory. And with some examples shown over here, one of the things that we didn't know what we would find is how much dynamic regulation of chromatin accessibility or peak accessibility would be found during differentiation. And some people would argue that you would find only these peaks that are progressively closed or those that are progressively opened, but we actually identified a lot of peaks that were transiently opened during differentiation, which was very reassuring. And some of those peaks were actually located and predicted to interact with transcription factors that define those states. We can also now, having this map of regulatory variation and gene expression in the same trajectory space, we are able to align enhancer accessibility, gene expression, and motif enrichment in the same trajectory space. And some things that emerged that were quite surprising to us was that in many cases, or maybe not that surprising, maybe one could argue it would be expected, that we can identify many enhancers whose accessibility precedes both motif enrichment and gene expression, particularly for the transcription factors involved in differentiation, suggesting that the epigenomic state of the epigenomic landscape of the genome perhaps undergoes more dynamic and regulation before gene expression changes. With that in mind, we wanted to revisit our question about the specification of excitated neurons in different areas. And towards this goal, we profiled single cells for RNA-seq and single nuclei for ATAC-seq for samples that were microdissected from either prefrontal cortex or visual cortex across 3 biologically independent human fetal samples. Performing this analysis with transcriptomics, as I mentioned before, recapitulated the patterns we've reported before. And as you can see over here, these are paired samples, in this case, a twin sample, paired samples of PFC and V1. And as you can see, the intermediate progenitor cells located over here are co-clustered across areas, but the excitated neurons develop into these distinct trajectories. Over here is a gene expression data for iomesodermine, the transcription factor defining intermediate progenitor cells. And as you can see, the area where it's most highly expressed over here is one where the transcriptomic identities from prefrontal cortex and visual cortex are shared. In contrast, when we looked at single cell ATAC-seq data, we found something slightly different, which was that we indeed still found that excitated neurons form these area-specific clusters. Here's data for 3 biologically independent individuals. But when we projected iomesodermine accessibility or predicted gene expression for iomesodermine, it actually fell into regions which were distinct across cortical areas, such that a branch point in trajectory analysis identifying the split point between prefrontal cortex and visual cortex actually occurs sort of right at this stage when the cells undergo this transient neural progenitor cell state. Using this analysis and this data set, we can identify peaks that are preferentially enriched in either prefrontal cortex or visual cortex cells and perform transcription factor binding motif analysis to identify those motifs that may be perhaps underlying these differential patterns of neurogenesis. So to summarize this part of the talk, we have sampled cells from distinct areas of the cerebral cortex, identifying the known cell types, including radioglia, intermediate progenitor cells, and excitated neurons. And while in transcriptomic space, these neurons or these cells differ across areas at the level of post-mitotic neurons, in the epigenomic chromatin state space, it is actually the neural progenitor cells that are already distinct. So with that in mind, what we hope to be able to do is to take advantage of some of these patterns of enrichment, particularly for transcription factors, to identify those that may be able to promote differentiation of neurons into distinct lineages. And one of the approaches that we envision taking is the in vitro differentiation from human pluripotent stem cells, which many of you know can be leveraged to derive distinct parts of models, in vitro models of the human brain, which is very exciting, using some very simple molecules that promote differentiation into distinct lineages. And in particularly taking advantage of 3-dimensional cultures, especially cerebral organoids, has become sort of this very popular way of modeling human brain. Using single-cell transcriptomics, we were able to confirm the identities that these cells and co-embedding with primary tissue cells, we were able to confirm that these in vitro differentiation states recapitulate many of the known gene expression profiles from primary tissue. Using transcriptomics and particularly these area-specific signatures, we are able to define a classifier which allows us to sort of recognize or predict the area information for every neuron sampled from an organoid. And as you can see from this analysis over here, the organoid differentiations can be quite variable depending on line and conditions used. So in the epigenomic state, we actually found a very similar pattern where while there was quite a lot of good correspondence between patterns of gene expression inferred from primary tissue and projected onto the map of organoid differentiation, such that radioglia cells, some clustered away from neurons, there seemed to be a lot more overlap between individuals and between conditions. Nonetheless, when we projected primary tissue-derived peak sets for every major cell type, we confirmed that these shared peak sets were consistent with predicted cell identity information. We then projected these peak sets.
This is pure
Yes.
just quick time check, 12:45 right now to kind of.
Okay, do I have 1 or 2 more minutes?
Oh, sure, yeah, just
Okay, great. Yes, I'm almost done. We projected these peak sets across, inferred from primary tissue across organoid samples. And something that was quite striking was that while some peak sets, cell type specific peak sets, were conserved across organoids, there was an overall sort of loss of fidelity of these cell types. And something that was quite remarkable was that many, particularly distal regulatory peaks found in primary tissues, were not conserved in organoids, as shared over here. And what was conserved between organoids was both the promoter and the proximal regulatory elements. Finally, in one last minute, I wanted to show you where we're going with this. And because of the tremendous amount of variability across organoids, we were wondering whether we can take advantage of our primary tissue resources to infer what signaling pathways could be used to harnessed to differentiate organoids into area-specific identities. And surprisingly, our analysis highlighted retinoic acid as a potential signaling pathway that may be promoting or regulating prefrontal cortex to visual cortex specification. And in experiments that I won't have quite the time to go into, we've exposed organoids to different levels of vitamin A and retinoic acid. And over here, what you can find is that organoids that were depleted for retinoic acid signaling pretty much all developed identities of the visual cortex, allowing us to sort of promote differentiation into distinct cortical areas. So with that, I would just like to summarize that I sort of emphasize the tremendous utility of single-cell genomics to both define normal differentiation patterns in the human cerebral cortex, compare those patterns to in vitro differentiation models, and perhaps start to learn lessons between the two. So I'm happy to take questions. Sorry for running long.
Yeah, thank you so much. So I'm going to allow people, you can unmute yourself right now if you have a question. Jill, you here, so I'll pass it over to you. So people can unmute, and you can also, I'll change the settings. You can, in a chat window, you should be able to chat to everybody and post your questions here as well.
Thanks, Piotr. It does look like there's a question from Alexandra. Albanese has his hand raised.
So should I open the chat window?
You can open the chat window as well. Yeah. But,
I was just clapping.
Tomasz, I was struck by of course, the difference between the fetal and the adult brain. And obviously it's hard to study kind of that path in between, but would be kind of essential in thinking about different therapy treatments and things for neurodevelopmental and neuropsychiatric conditions, since there may be different types of cells present or different expressions. Can you comment on how, I don't know, your work may attribute to to that part of.
Yes, thank you so much for the question. So as you say, the human development is quite long and surprisingly, or maybe annoyingly so in some cases. Those of you who have children certainly know how I feel about that. Indeed, what's missing from the picture is sort of the third trimester. However, working with, I think it's not true that it's impossible to access some of the difficult to access samples, particularly for the early postnatal, neonatal samples, working with local collaborators at UCSF. We are indeed able to access some of the early postnatal primary tissues, and we are now extending this analysis to those stages, particularly stages of early postnatal life, when when some of the early onset neuropsychiatric disorders can be found. And in many cases, it is a difficult challenge because it requires a lot of coordination across the clinicians, the hospitals, all the regulatory sort of bodies. What's challenging, of course, is that for some of those disorders, particularly autism spectrum disorders, many of those genes that are implicated through human genetics studies, their gene expression profiles are actually enriched prenatally during fetal stages of development. And so we think that some of the patterns that we're recognizing right now are actually revealing some of those putative selective vulnerabilities, which could be modeled functionally using models such as cerebral organoids. Of course, it poses another challenge, which is that unfortunately there are increasing restrictions on the use of human fetal tissue for research, and that poses yet another set of challenges. So it's a difficult topic, which certainly lacks suitable experimental models, but is important because many of the psychiatric disorders currently have no treatments whatsoever, and yet they affect so many children, and there is no cures.
Thank you, and really beautiful talk and work. There's a question in the chat box.
So the question is...
so Satar, I'll unmute you if you wanted to ask directly right now.
Hi, yeah, great talk, very interesting. So, you know, one thing I've been sort of trying to figure out is, you know, with single cell technology, sort of one of the challenges is obviously yield and cost. And, you know, with single, say, ATAC-seq, sometimes it may be, in my mind, even harder to sort of filter out you know, droplets that have more than one nucleus in them. So I was wondering if there are any strategies you've used to sort of increase yield, maybe super loading, and then later on using the computational techniques to sort of remove the outlier cells. Yeah.
Sure, thanks. That's a great question. Yeah, as you say, multiplets are something that we're extremely worried about, and we've really not addressed that question extensively yet. What we're doing right now is we're taking advantage of some of the cholesterol modified conjugated oligonucleotides, which allow you to actually multiplex nuclei into single cell ATAC-seq approaches. And that hopefully will provide us with a data set that would be a useful training data set for deep learning algorithms that could then allow you to sort of distinguish between doublets and singlets in this analysis. We've not yet tried to superload, I think partly because of that concern that the more you superload, the more you lose in terms of the potential running into the risk of running multiplets. But I think going forward, seeing the amount of variability between samples and also between individuals, I think it will be tremendously important to be to think very seriously about running multiple individuals or as many individuals as possible on the same day, taking advantage of some of the multiplexing strategies that are becoming. Of course, for humans, it's a lot easier, right? Because you have SNPs, right? And so one of the things that people are currently working on is taking advantage of the fact that humans are so variable. And by looking at individual SNPs, you can sort of re-identify. And that's a lot easier with single-cell ATAC-seq than it is with single-cell transcriptomics, where there are fewer SNPs to do that efficiently.
Okay. Any other questions? Xing, we can unmute you right now.
Nice talk. So it's interesting to see that epigenomics, you can use it to predict the cell fate. So just wondering, like for RNA-seq, is there anything that RNA-seq that cannot see, but only the epigenomics, such as ATAC-seq, can see in order to determine the cell fate, or basically with RNA-seq, with those transcription factors, you can still use it to predict?
That's a fantastic question, and I've been thinking about that problem for a long time. One of the challenges that we've seen with single-cell genomics especially is that the epigenomic information typically doesn't recognize as many clusters. And so, do you really need to do epigenomics at all? And something that we found in our work is that when it comes to development and when it comes to recognition or diversity of neural progenitor cells, especially as I've shown in my talk, the sort of areal identities of neural progenitor cells are highly distinct between prospective functional areas. With transcriptomics, we don't see that divergence. So for some reason, and it may be as simple as the number of features that distinguish these two progenitor types located in different regions, the epigenomic information allows us to recognize a further diversity within a previous transcriptomically similar cluster. Now, when it comes to the transcriptomic question, one of the central questions in my mind is, well, we have the transcriptomics, could we just predict the transcription factor code from transcriptomics? And there are a number of problems with that approach, or a number of challenges. And one of those challenges that is very fundamental to the basic biology, which are twofold. One of them is the fact that transcription factors are typically not expressed for a very high number of molecules. So for many transcription factors, maybe not all the developmental ones, sometimes they're present in single molecules. Whereas if you're thinking about the epigenomic space, you're suddenly looking at motif enrichment that's distributed across hundreds, if not thousands of peaks. And so purely by sort of flipping the equation of looking at a single event, the transcription, or trying to detect a single event, which is the expression of a transcription factor, versus being able to sample through all the epigenomic peaks or open chromatin peaks that are enriched for that transcription factor binding event, you're increasing the power of being able to reliably detect or predict transcription factors that have been acting on that cell throughout development. And then the other problem, of course, is that in many cases, and we've not yet seen evidence for this with ATAC-seq, but we've also not really looked, which is that many people sort of would argue that the epigenomic state is sort of a more stable readout of cell identity. And even if the transcription factors are no longer expressed, you may have a stable reorganization of the epigenome that's present there. And that was very beautifully demonstrated by Joe Ecker's group, looking at non-CG methylation, where regions of the chromatin that were hypomethylated near transcription factor binding sites that were only present during development, suggesting that the epigenomic state has information of the historical information of the transcription factor binding events that were present previously. We've not addressed this yet with single-cell ATAC-seq, but it's certainly something that I think eventually will prove to be true, that the epigenome has a lot more, it's more of a tape recorder for cell identity. And even if you no longer recognize a transcription factor, the chromatin state is a reflection of what happened in the past, if that answers.
Yes, thank you.
So Alexander, you have a question? I'll unmute you right now.
Given the loss of fidelity in the cell types that you mentioned with the cerebral organoids, I mean, what's your take on how useful they can be for polygenic or epigenomic factors underpinning disease? Do you think that they might just lead to artifacts? Do you think they're still somewhat useful?
No, that's a fantastic question. And I think, I'm a big proponent for organoids. I think they're a fantastic model because for the first time, we're actually able to model genetic disorders using CRISPR and whatnot. They're useful to some extent. They're very useful at modeling cell classes. So broad cell classes seem to be fairly well recapitulated. But when you want to get into distinct cell subtypes, I think they become a lot more problematic. What the underlying reasons are, I don't know. We have hypotheses about there probably being remodeling due to metabolic stress that they experience in a dish. And it is possible that with some of the more sophisticated bioengineering approaches or culture modifications that will get rid of some of this stress, we will actually allow these cells to differentiate into sort of higher fidelity models. But right now, certainly there is a concern that we're pretty good. And whenever you look at any organoid paper, you typically say, well, there are excitatory neurons and there are inhibitory interneurons, and therefore cortical development is solved. Well, I think that's too simplistic, that there are many subtypes of cells that exist. And while you may be able to use them with the current state to address some broad questions about cell classes, I think we still need a new generation of technologies that will increase their fidelity before we start accessing the more refined cell types and start asking the more refined questions.
Thank you.
Well, I guess that brings us to 1 o'clock. Doesn't look like there's any final questions. So I just want to take the minute to thank you very much for your presentation and sharing your beautiful work with us today.
Absolutely. Thank you so much for inviting me. And if anybody has any questions, please feel free to reach out. Happy to answer. Stay in touch.
Yeah, pleasure.
Bye.